Hello, everyone. Welcome to our monthly Science of All Protein seminar. My name is Bianca Dada, and I am the Scientific Partnerships Manager at the Good Food Institute. For those of you who are here for the first time, welcome to our community. The Good Food Institute is a nonprofit think tank 100% driven by philanthropy, and our goal is to build a world where alternative proteins are no longer alternative. If you're looking to take a deeper dive into the science of plant-based, fermentation, or cultivated meat technologies, check out our open access resources at gfi.org/science. Today, I'm really thrilled to be joined by two amazing PhD students, Jorge Guadalupe and Anna Thomas, who will be with us to discuss their research projects on cultivated chicken scaffolding and large language learning models. With that, I'd like to introduce today's first speaker. Jorge Guadalupe is a biologist with a master's degree in cell biology from UFMG in Brazil. During his master's research, he developed a thesis focused on scaffolding for cultured meat and contributed to the creation of the first cultivated chicken steak in Latin America. He is currently pursuing a PhD at UFMG, where his research centers on hydrogels for 3D bioprinting of cultivated meat. In addition, he collaborates on several projects with professors Andrade and Jorge, including work on cultivated fat, cultivated leather, bioreactor design, and microcarrier development. Recently, Jorge was awarded a Fulbright scholarship to carry out a project at the Kaplan Lab in the field of metabolic engineering for cell growth optimization within the cultivated meat bioprocess, which we're really excited about. Jorge, thank you for being with us today, and I'll turn the floor over to you.
Thank you, Bianca, for your warm introduction and for all of the GFI team also for the invitation so that I can share my work here with you. Hi everyone, I'm Jorge Guadalupe. I'm a PhD student on cell biology and bioengineering at the Federal University of Minas Gerais here in Brazil. And I'm going to discuss with you a little bit about the work that I've developed on my master's that's called Engineering Cultivated Chicken, Exploring Mechanobiology to Reduce Costs. Everything started once again with GFI actually. So GFI was the first spark at UFMG in 2021 when we got this research project that's called Hybrid Structure Scaffold to enable the production of chicken muscle with fat for cultivated meat. This was a very extensive project that was led mainly by Dr. João Paulo Santos, Dr. Aline Brunner from Cefet, and Dr. Luciana Andrade, my advisor, and Dr. Erika Jorge from UFMG, and all of the students that were in the bench doing the experiments. This is me. The general strategy, actually, the project of this project was to create a scaffold, which is this three-dimensional structure where the cells will be plated upon to create a three-dimensional muscle after plating the cells. But instead of only having the fibers that would mimic the extracellular matrix, we would want to apply canola oil albergated in hydrogels to create a hybrid hydrogel. Why is that so? So first, why did we wanted to create why did we want to use cellulose acetate nanofibers? Because they were very similar to the extracellular matrix, very much more related to the width of collagen fibers in nanometric width. And it's very easy to produce by electrospinning, and it's very biocompatible. The cells love it. And why the hydrogels microparticles? Because we wanted to add fat without the need of adipocyte and muscle co-culture. And the canola oil is already an oil that is used on plant-based industry already, because it mimics very well the fatty acids of conventional fat. So the whole strategy of the project, after we have this nanofibers, would get satellite cells from chicken and recreate this structure cultivated chicken with both plant-based oil and the cultivated muscle itself. And what we wanted to do was divide this project in three different branches. The first branches would be in Cefet, where they would produce the hybrid scaffold because they have a lot of knowledge on material sciences. So they would produce the nanofibers, the hydrogels, and then the combined hybrid structure. At UFMG, we would first validate what is made on Cefet with C2C12 myoblasts, because simultaneously there would be Dr. Erika's Jorge lab designing all of the experiments to have the primary chicken satellite cells culture validated and maybe immortalized. So the whole master's that I had actually was focused only here on the validation of C2C12 and on the cellulose acetate nanofibers. I'm gonna delve into the other fat results as well at the end, but like all of the things that we found out with this nanofiber were very exciting and enough to publish a paper. So this is the paper that we published in 2023, 2024. I published this together with Anelisa Antunes dos Santos, a former PhD student at my lab. We called it Random Cellulose Acetate Nanofibers, a breakthrough for cultivated meat production. And here we delved into the comparison between random cellulose acetate nanofibers and aligned cellulose acetate nanofibers as well. This is easily done by electrospinning by different speeds of the collector. So if you make a high speed on the collector, you have aligned mesh. And if you slow it down, you have a random mesh. And why did we want to investigate both? Because usually on literature for skeletal muscle engineering, they prefer to use scaffolds with topographical cues because it will guide the cell's alignment, which is something that we want a lot so that the cells mature and be fused, multinucleated, and with fibers inside, which is the final maturation process of the cells. And very different from the random one, which has no topographical cues. However, the main aim of a scaffold on any type of tissue engineering is to mimic the extracellular matrix of the native tissue. And this is the skeletal muscle extracellular matrix. So it's much more random than the aligned mesh. So we wanted to make this analysis as well. The first result that we had was on adhesion. We had this control group on a monolayer and a two-dimensional culture to see how the cells behave very, very differently on the two-dimensional environment. So here we have a cells flat and kind of very well spread. And here we have cells that are actually a little bit elongated, a little bit stretched, both on the random cellulose acetate nanofibers and the aligned cellulose acetate nanofibers. And this is only 6 hours after seeding. Something that we also came to our eyes actually was that you see a lot of these structures that look like phylopodiums of cells that are that has the cytoskeleton very active. I'm gonna call this attention right now because this might be useful for understanding some other results later. And going on to viability and proliferation, we made this analysis on 3 days on growth media, which is DMEM, 10% of fetal bovine serum. And we saw that the cells were very much alive on both scaffolds. Here you can see that the aligned one, because of the topographical cues, cells tend to be a little bit more aligned than on the random one, but they're both very viable. And trying to guess a little bit of proliferation, we performed an MTT result. And here on the monolayer, which is our control, we could see that the cells would increase its proliferation, its viability on the first day of culture. And on the third day of culture, you also see this increase, which is not seen on the aligned cell, on the aligned fibers, and on the random fibers as well. So this reduction of viability here on 72 hours could be cell death, but on here on the live and dead, we don't see relevant cell death. And on stem cell biology, either the cells are proliferating or they are differentiating. So if we see that the cells are not dead and we see this reduction on the bar, very likely the cells could be differentiating on the scaffolds, even though I'm putting media to stimulate proliferation, which is DMEM, 10% of fetal bovine serum. So we aim to investigate a little bit of this differentiation process. And regarding the differentiation process, we have different morphological changes, as I've mentioned already, on skeletal muscle. So one of them is like having, this is a quiescent satellite cell on a native muscle tissue. We call it quiescent because it's like sleeping, and when there is something that activates it, we see this proliferation, mitosis happening. And after there is an induction of differentiation on the body, usually inflammation cytokines that can induce this, or even matrix remodeling, induce this differentiation. There are a lot of chemical stimuli inducing this differentiation, and on in vitro, we have a reduction of the serum. Instead of having 10% fetal bovine serum, we have a 2% horse serum, so that we diminish the availability of growth factors, and the cells will start to morphologically change, so be a little bit more elongated, and then starting to fuse with one another, forming myotubes, and then forming the myofibers as well, where we have the complete protein apparatus for the contraction as well. For that, we stain the cells with phalloidin that will stain the actin cytoskeleton. And then we can see here on the aligned nanofibers that we have this pattern here on 72 hours where the cells are starting to align with each other and starting to form structures that really looks like multinucleated cells. Both in the aligned and in the random one. We can see these structures on the random one as well, although they are not completely aligned with in one direction. They have different directions as well. So this gave us a glimpse that if the cells are differentiating, maybe they are here between this process of myocyte and myotube. So for making a little bit more investigation on the morphology, we did this scanning electron microscopy, growing the cells 10 days in growth media, DMEM, 10% of FPS. And we can see here, even on the random cellulose nanofibers, the cells tend to make some sort of alignment. And when we make this approximation here, this magnification here, we can see that the cells are a little bit chubby because it looks like they are fusing with one another. And even with growth media. And actually, on the random one, cells look a little bit more differentiated than the aligned one, where the cells are a little bit flat here. You can see this huge cell here, but here on the side, they are a little bit flat. But we wanted to compare with the perfect differentiation group, which would be when we induce chemically the differentiation, which is differentiation media, DMEM 2%, Horth serum, as I said, reducing the growth factors availability. And it's quite similar. We don't see like a lot of difference on the morphology. In fact, I hope I see that the cells are actually more, it looks they are more differentiated here than here, like because you have more structures that of this multinucleated that looks like chubby cells. But we cannot say that it is a complete truth until we go to the next step, which would be delving into gene expression of the cells. So just like there are some morphological features that address this myogenesis, we have also molecular features, such as these different waves. Usually a quiescent satellite cells is expressing a lot of PAX7, and there is a reduction when activated. You have a first wave of MyoD, then a second wave of myogenin, and then a wave, which is not a wave, it's a plateau of myosin heavy chain, which is the actual protein that will make the cell contract. So because we thought we were here on myocyte and myotube, we made this, we chose myogenin and myosin heavy chain to check with protein expression, RNA expression actually, qPCR. So first here in blue, we have the results of myosin heavy chain 3 and myogenin on aligned cellulose nanofibers. And what we can see here is a qPCR is the relative expression in the qPCR related to the first day. So what we have here is like we don't have any difference on the second day of level of expression regarding to the first day. But on the third day, we have around 20 times more expression of myosin heavy chain on the third day related to the first day, okay? And so we see this increase in growth media in the third day, in the 10th day as well. But when we make this comparison, this empirical comparison between 10 days with growth media and 10 days with differentiation media, we don't see like this much higher standard here on the differentiation, showing that it looks like the cells are actually behaving the same way in the growth media and in the differentiation media. And here on the myogenin as well, we see already this higher level of expression, around 7 times more expressed on the second day regarding to the first day. And in the 10th day, although it looks like there is a reduction here, we didn't see, because of this big standard deviation, it's not like that relevant. We can call it a reduction. But we do not see many differences on the 10th day of differentiation media and on the 10th day on the growth media as well. This is the aligned one. When we go to the random one, we're not talking about 10, 20. We're talking about 500, 1,000, 200 times more express these two genes. And just like that, in the 10th day, differentiation media and growth media, they look very much the same. So we did see a huge expression of these genes in growth media, even when the cells are not being induced, therefore, with no differentiation media. So the cells are actually differentiating in growth media. And we wondered, how can this happen? But before we did this, all of these experiments as well with the chicken cells, and the chicken cells also showed high alignment. This is the random cellulose nanofibers. The cells showed high alignment, and this is also showing on the scanning electron microscopy. This really looks like a chicken. Look, the texture. But wondering like how could the cells differentiate even when not stimulated with differentiation media? We stumbled upon this very interesting result, which is the mechanobiology. So cells are able to sense chemically the environment, which is like all of this intercellular cascade that we are, how medicine work. They really delve into this. So the has a receptor. The cell has a receptor. This receptor will find the its ligand, and this will induce some sort of modifications on this protein that will induce this intracellular cascade that will lead to protein expression here at the end. However, cells are also able to sense mechanical stimuli, such as stiffness, strength, tension, compression. So who senses this is the actin cytoskeleton mainly. So the whole cell cytoskeleton, but mainly the actin cytoskeleton, because it's very peripheral, the actin cytoskeleton. And this will lead, this is like a wave like on a rope. It's much more complex than this. I'm going to show on the next slide. But it leads to the nucleus. So it's not a wave on a rope. It's a lot of intracellular proteins that will lead to this. It's not completely elucidated. There are many pathways on intracellular, what we call mechanotransduction. So the cell sense something that is mechanical and will induce this as protein expression that will, so we will transduce this into chemical signaling. So you have tension, substrate stiffness, compression that can be felt because the cells are attached. The actin cytoskeleton is linked to the extracellular matrix through these proteins that are mainly orchestrated by integrin. And this will lead to very, very different types of intracellular cascade that are not completely elucidated, that will delve into gene expression. One of these intracellular cascades is the IAP-TAZ or hyposignaling pathway. So IAP is a protein that is on the nucleus when it's activated. And when it's outside the nucleus, therefore it's phosphorylated, it doesn't go into the nucleus and it's inactivated. And there are a lot of very interesting results that show that when IAP is inside the nucleus, it targets a lot of genes related to proliferation. And when YAP is outside the nucleus, it does not stimulate proliferation and therefore could lead to some other type of cell behavior, such as its differentiation. On muscle cell biology, it's really well known, such as I said, all of this blue genes, the waves of MyoD, the reduction of PAC7, the next wave of myogenin, it's very well known, very well described. But it's also very well described IAP expression on muscle. So when you have muscle injury, you have recruitment of satellite cells to be proliferated to make a new muscle so that this new cell will be fused with your muscle and create this new cell that was injured. So we know that we also know that when you have this sort of inflammation at the beginning, you have some extracellular matrix remodeling, increasing the stiffness of your muscle. And it's known that this increase in stiffness stimulates IAP to go into the nucleus. This is what happens. And this will stimulate proliferation and cell expansion. And when you have this later remodeling of the extracellular matrix, going back to the initial stiffness of the muscle, which is around 12 kilopascals, you have an increase on phosphorylated IAP. Therefore, IAP goes out of the nucleus and goes to the cytoplasm, stopping to induce the proliferation. It's very well known this. So we wanted to see if this is happening on our system as well. As I said, if you plate one muscle fiber on 1 kilopascal, which is the stiffness of a muscle of an adipose tissue, it does not make the striations that is very well known on muscle or the contraction. This is striations. But when you have here at around 8 to 12 kilopascals, you see these striations. When you increase the stiffness, you do not see this striation. So it's really well known that skeletal muscle native elastic modulus, which is around 12 kilopascals, stimulate myogenesis. To see what is the stiffness of our nanofibers, we did this experiment with atomic force microscopy, where we can sense the elastic modulus of we call it Young modulus, of our mesh of nanofibers, depending on the type of the tip of our cantilever probe that we have. And what we saw is that in both aligned cellulose nanofibers and in the random scaffold as well, we have a stiffness of around 12 kilopascals as well. Very interesting. So to try to understand if it's really YAPI that is related on this process, we check this on immunofluorescence of YAPI and on the monolayer, on the random cellulose nanofibers, and on the aligned as well. And we counted how many nuclei were positive for YAPI. Therefore, if they're positive for YAPI, they're stimulating myogenesis. And if they're negative for YAPI, they're not stimulating proliferation if they're on the nucleus. And if they're not on the nucleus, they're not stimulating proliferation, very likely inducing the differentiation as well. And we saw that on the monolayer, which is a glass cover slip, gigapascals, we see that we have 80% of the nucleus positive for YAP. When you go to the aligned one, we have around 2% of nuclei positive for YAP. And on the random, we didn't have any one on 3 experiments, different experiments. So it really looks like there is a process of mechanotransduction related to YAPI on our system. This is very interesting because it means that we have one scaffold that stimulates the differentiation regarding of media administration. And because media, very well known, this is a report from GFI, it's a little bit 2020, like it's not as new as some other data, but it still highlights how difficult it is, even though in different types of scenarios, the cost per liter of different types of media could be very, 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 very expensive. And this is one of the main cost drivers. And also one of the main cost drivers of cultivated meat is the time, is the energy you're gonna waste, you're gonna use on the cultivators, on the bioreactors. And the more time you need for differentiation, which takes around 2 to 8 weeks, which means 2 months inside the bioreactor, maybe we can reduce this time. It doesn't mean that we don't need to administer the differentiation media, but maybe coupling the scaffold that induces the differentiation itself with the differentiation media, you can reduce this to 1 week to 2 weeks. We need a lot of new research on this upscaling, the scaling up of this process, but this is a very interesting result that we had. And differentiation matters because if we have differentiation, this is where you're going to have protein content, the myofiber formation, which means the nutrition and also the taste and the texture, the cell alignment, and all of the protein inside it. And talking about the taste and texture, not only protein is made, but also the fat content. As I said in the beginning of the project, we had this big project where we would put the canola oil inside of hydrogels to make the hybrid scaffold for cultivated meat. We did it. So this is the alginate microcapsules containing the oil, and we did these strips so it would prevent even more having like the second layer of alginate in the strips that would contain a lot of this alginate microparticles to prevent oil spilling to the scaffolds. This is the strips of oil, and this is the cellulose acetate scaffold. And we plate them with chicken cells, and the cells were pretty much viable even when compared to the control group, showing that this is very feasible for cultivated meat as well. UFMG didn't stop there. So Dr. Erica Jorge's group actually thought that what if we take the cellulose nanofibers that were like amazing for muscle cells, and we also put adipocytes to make cultivated fat. And they did it. The cells, this is, you can see here on green that would stain lipid droplets, would also stain the nanofibers. You can see a lot of fat content on the cells. Here also as well on the scanning electron microscopy, you can see a lot of these adipocytes, showing that the cellulose nanofibers can allow pre-adipocytes full differentiation and support long-term culture. and they do not float when cultivated onto the cellulose acetate nanofibers, because one huge drawback of cultivating adipocytes would be that when you have the differentiation of these adipocytes, they would float, they would stop the adhesion on the whatever substrate that they have, and they would float. This would be very difficult to be retrieved on a bioreactor. And we also used this technique of stacking that we developed to stack together muscle cells and adipocytes. And we made this very interesting. This is unpublished. This is going to be published very likely on this, hopefully next month. But here on red, we have the muscle cells, and on green, we have the adipocytes. These are all chicken cells. We had this different, also another showing another image showing them two together. And here on the Z as well, on the thickness, you can see that we have some thick tissue that could even be imaged here to show very aligned cells and a lot of fat deposition as well. This is our cultivated chicken muscle, published result. This is our cultivated chicken fat, published result, and this is the unpublished result on first hand, showing to you our cultivated chicken meat with all together muscle and fat here developed at UFMG as well, even fried here that we had. This is it. I'd like to thank GFI once again for the opportunity of sharing this work. All of my research group, once again, to the professors, Luciana Andrade, my advisor, Erika Jorge, and Aline Bruna, and all of the team here at UFMG Alt Protein Project as well. Once again, GFI here. These are not only students that are making events on campuses, they are researchers. All of them here are involved a little bit in one of these results as well. Also, thanking to the university, to the program, to all of the funding agencies, GFI being one of them, and also highlighting that here at UFMG we have one baby step program that's called the Center of Technology on Cellular Agriculture, that we hope to increase a lot of our great data as well. Thank you very much, and I'm open for questions.
Thank you so much for that lovely talk, and I really appreciated how you outlined the different research questions that came up for you at each point in that research journey, and then the methods that you used to answer that. So thanks for making it so clear to follow and bring us along for the ride. It's also such a joy to hear about kind of how you've interacted with GFI at different points in all of these stages of your research. So thanks for sharing that as well. I have one question I'd like to start you off with, which is about how to tune these scaffolds. So, is there a way to potentially create scaffolds that are tunable for different phases of what you might want from kind of this cell growth, proliferation, and differentiation, potentially in response to different environmental cues, or is that something that you've looked into at all?
Well, this is a very good question because this is my first question of my PhD. What I wanted to make was a hydrogel that would be stiffer in the beginning so that we would stimulate cell proliferation, and then somehow like a tunability, a temporal tuning. Somehow I could decrease this um, stiffness. This is very difficult. Like I gave up on this on my PhD because it's very difficult so that you can control this on a temporal level. There are some results that show this, but mainly with hydrogels. I've never seen this with nanofibers, as far as I've seen. But it's a very interesting topic. I showed you that you can tune the scaffolds, but it's prior cultivation. So like when you start the cultivation, it's much more difficult so that you can tune this. I cannot foresee anything that could right now make this temporal tuning, but it's a very good topic as well.
Awesome. We'll have to keep dreaming about ways to figure that one out, but really cool to hear that you thought about that. We have a question from Ryan about the cultivated chicken tissue and the unpublished result. Was this tissue generated via co-culture, and can you provide a little bit more detail about the methods that you used to generate that sample?
Yeah, sure. That's a very good question because co-culture is one of the main drawbacks, of the main bottlenecks on cultivated meat. We did some different types of settings on this data. So we did one setting that we would try to see the cultivated tissue together. After culture, so we made the differentiation of each one of we had like this meshes of nanofibers. We would plate the muscle cell here, we would plate the adipocyte here, we would induce the differentiation here, inducing the differentiation here. And then there was one setting, I don't know which one of the settings will go to the paper, okay, but I'm gonna discuss the backgrounds of research. There was one setting that we did this and let them for 2 days so that we would attach with one another in if I'm not sure, if I'm not wrong, the differentiation media of muscle cells. So we kept them on DMEM 2% on all of them together, the adipocytes and the muscle cells. And we also had one setting that we would only see if we could image them together as well. So we had them separated and we placed them one above the other so that we can make the imaging on confocal microscopy just to see if it worked. Both of them worked. What I'm showing to you is this one that we left for 2 days or 3 days, I don't know, together with one maintaining type of media for cultivated muscle. Sorry, my broken English.
That's perfect. Thank you. Very fascinating. And I am very much looking forward to reading that when it's out. How are you thinking about the potential implications of this study on other cell types? So obviously, chicken is really appealing for a lot of reasons, but is there something unique about chicken cells, or do you see this translating well to other animal cells?
Well, that's an interesting question because we could see that even using C2, C12 with some urine cell type, we saw this differentiation. And when we used poultry cell type, we saw this differentiation as well. So we are really looking forward so that it's not species related. It's myoblast related. So like if we plate a myoblast, and there are some differences on this is a thing that came up to my mind because I'm not sure how are the stiffness of all the animal relevant species for cultivated meat. This is something interesting, but I'm sure it will not differ a lot on the I don't know, from 1 kilopascal to 20 kilopascal. It might be around there. So if you can induce this, and this is something that I know that we can do depending on the speed of the deposition of electrospinning of the fibers, we can make a scaffold that maybe is like 12 kilopascals, 15 kilopascals, 8 kilopascals, and then we can tune this for different species. But I think it's because we saw the same result with poultry and with mammals, it will keep that way.
Amazing. Okay, that's great to know. Thank you. We have a question from Sergio. You mentioned that the cost of the culture medium, you mentioned the cost of the culture medium and noted that this remains one of the major challenges for cell cultivation. With that in mind, can you share more about the main sources of proteins and amino acids that you used in the culture medium to enable the cells to synthesize myofilaments? And also, do you believe that these results could be replicated without the use of FBS?
Ah, this is a very good question. The question of $1 million. Well, the amino acids that we used were from the DMEM. So this is a lab scale, just to make sure. Like, this is a validation that we wanted to make on the hybrid structure. So like dividing into small steps so that we can come into the end of the day that we want to recreate a skeletal muscle tissue, which is not an easy task. But like on the first step that we had was the creation of this hybrid structure. We did this hybrid structure. We validated this hybrid structure. So we didn't focus on solving the issues with culture media. There are different research projects on UFMG trying to solve that as well. But here we didn't try to solve that. So we use the DMEM, which is like almost all of the amino acids are present. It is the pharmaceutical industry DMEM that we are using, and to see if it would behave very well with a culture without, for example, an essential weight media or some sort of replacer on FBS. We didn't go into the scaffold itself, but we have some results showing that the cells can behave very well with reduction of FPS even chicken cells, reduction of FBS, and increase of some other supplements such as insulin, transferrin, and other media supplementation that we have. We saw this in 2D culture, but it would be very interesting to see this on 3D culture on these scaffolds as well. So I think it's not an easy task, but I think it's absolutely feasible. Yes.
Like you said, it's $1 million question, and I think it's going to be a really interesting one for you to discuss with some of your new colleagues at the Kaplan Lab, because I know they've been doing a lot of work on thinking about things in this space. So it's going to be a lot of
Definitely.
fun for you, I'm sure. Another question from Camilo. Are you planning to do any kind of techno-economic analysis of this process to foresee the scalability?
Well, this is an interesting type of talk of from GFI because it's PhD students doing the presentation. I intend to do a lot of things, but after I graduate from PhD and I start my own research group, who knows? I don't know. But like right now, this is a great question for my advisor, Dr. Luciana, if you're watching. This is something interesting to delve into because um, I think, but this is like a huge guess. I'm not completely backed by data, but it's really promising if you have a scaffold that induces the differentiation itself. We need to see how much time we're going to reduce on cultivation or how much media we're going to reduce on cultivation. But this result highlights that it's very possible that it can reduce a lot. How much? I don't know. And a technical analysis, technical economic analysis would be very interesting to delve on that. But we would need collaboration. If you know how to do it, reach us.
Love that. I love that call for collaborations, and it's definitely something we want to encourage. And I also love that you're giving us a mini teaser of what your future career could look like. So fun to think about. Another question from the audience. Do you also do the metabolomics and vitalitomics of the cell scaffold that you developed and look into whether it mimics a similar profile with regular chicken fat?
Yeah, this is a very good question. There are students doing this. There are two students from Dr. Erika Jorge that are delving into this research, this type of question. I don't know their data yet. I think they're gonna be published soon. So keep on track on YFMG Out Protein Project LinkedIn. We share all of the researches that we've published there.
Fantastic. Thank you. We'll stay tuned. I have a question for you about kind of the nature of the work that you did here. I think one of the things that's so impressive about the study is that there's so much breadth, and some of that seems to have come about due to this collaboration of these three different groups together. So, can you speak a little bit about kind of how that collaboration came to be, and then also what you've learned from kind of navigating doing science across multiple different labs and bringing those together and figuring out how to kind of hand off results to each other.
This is a very good question, actually. So, all started with Aline, which was the researcher, the principal investigator. She was a professor at Cefet, and she had a startup uh, that was accelerated on the UK, and they like in 2027 they were one of the first that was discussing cultivated meat. And they had this researcher, which is Annelise, that was the co-author of the paper with me that wanted to make a master's on cell biology. She wasn't a materials engineering. Aline was a physicist. They had the scaffold, and they wanted to validate the scaffolds with cells. So they knocked on doors, and Erika and Luciana opened the doors. So that this is interesting. Let's try to see this because they had their opportunities maybe on other type of tissue engineering, not necessarily skeletal muscle tissue engineering for cultivated meat. But then the doors opened once again with GFI with this research project on that RFP from GFI in 2020 for 2021. And they decided to write this paper down. And the whole strategy was to make like this once in every 2 weeks we would make a meeting, and one of the students would present their uh, progress from that time. And it was something that was very interesting because like for me, which I was really young, I was like 22, so I learned a lot on time management, on teamwork, because like I needed in order for my results to work, I needed the scaffolds to be well produced and validated on Cefet, on the material science part. And in order for the chicken results succeed, they needed my results per year. So it was a really work team. And the last thing that I learned a lot and that was very great for me was the interdisciplinary, because like I learned a lot of the results from the material science, which is something that I'd never even stumbled upon during my bachelor's. I had this opportunity during my master's. So I think the interdisciplinary feature was the main one.
It's
I don't know if I
incredible to
answered completely
hear.
the
No,
question.
that was lovely. Thank you. It's actually really incredible to hear about all of the different um, scientific perspectives that came together to make this happen, and really helpful to
Absolutely.
kind of hear the different curiosities that led to this moment. So thank you for giving us that
Yeah,
insight into the
of course.
process.
And just like a keynote, because like cultivated meat is there are a lot of bottlenecks yet. So if you don't have this interdisciplinary approach, we don't solve anything. This is why I'm looking forward to learn a little bit about artificial intelligence with Anna.
What a lovely plug, and I'm really looking forward to hearing what Anna has to share as well. I think we have one more question for you before we transition over to our next speaker, from Tuna, asking about the test for MHC3 and myogenin, noting that the measurements were taken at 3 days and 10 days. So the results for both time points looked kind of similar. Can we say that this indicates the plateau that you mentioned in relation to myofiber formation? And does that mean that the myotubes are already formed by day 3?
Well, myotubes are formed and on scanning electron microscopy. Myofibers are not formed on day 3. And it's very difficult to have a complete differentiation system on an immortalized cell line because they are immortalized. And usually, when you immortalize the cell, you kind of lose this type of system that will trigger its completely maturation because it's prone to proliferation. So it's a tricky thing that we'll have to figure it out on genetics for cultivated meat. I cannot say that it indicates a plateau because um, some data from other papers, just like I've shown as well on the scheme that I had, was like the differentiation process for cultivated meat can take from 2 to 8 weeks. So 8 weeks are much more than 10 days. So very likely this is a growing slope. So it's very likely that even though they are similar on day 3 and day 10, maybe on day 40 there will be an increase and then we'll have a plateau. So we would need a time-dependent experiment that we would see long-term culture of these scaffolds and how the cells would behave on this. But this is an interesting question.
Amazing. There's one follow-up question on that from Julia. Based on the protein expression over time and the muscle growth phase graph, it seemed like you would expect that the myogenin would increase first and then decrease, while the MHC3 would increase later. But it looked like they both increased and then plateaued at a similar rate, although at different levels. So does that give us any more insights into the stage of differentiation?
Julie, this is an absolute lovely question that I've had during all of my analysis of qPCR of this experiment. Because, again, I don't think it's a plateau. I think to say it's a plateau, we would need a long-term culture. But things are messy. We're talking about on the cytoplasm. We're talking about RNA expression. Like to delve into the actual function, we could do a Western blot to see protein expression to see if we actually have the MHC. And like before having, although we have these molecular waves, the assembly of these proteins and the function of these proteins, they behave differently. So something that I discussed a lot with my advisors with this in all of this meetings that I told you that we have with this project was that it can be messy sometimes, and it's okay that it does not make 100% sense to so that you can have the perfect wave of the book of the you know the classroom book of myogenin and then a plateau of myosin heavy chain. So we can see what I got from this data is that I can see that there is an increase on differentiation, mainly on the random cellulose fibers through the completely higher fold change that we have on the random one, talking about 100, 1000 times more expressed than to the aligned one. But like on the actual stage of differentiation, it's hard to say only with these two genes and this time frame that I had.
Thank you for that, Georgie. I really appreciate all of your in-depth answers to this, the curiosity that you brought to it, and I really want to uplift one of the comments that you made about the need for interdisciplinary work. So definitely looking forward to hearing about how the next phase of your research goes and staying tuned for the publications that are coming out of this work. Thank you again for sharing that with us.
Of course, thank you.